Welcome back all of you. Myself L.Q. Rehman. Dear students, today we are going to study the enzymes involved in DNA replication. Before that, we need to study the basic biology of central dogma. Central dogma was proposed by Francis Crick in the year 1958. He describes the flow of genetic information in living cells. According to this, genetic information flows in a unidirectional manner, that is DNA, DNA to RNA, and RNA to protein. There are three major key processes in central dogma. First one is replication, second one is transcription, third one is translation. What are the significance of it? It explains gene expression and protein synthesis, which forms the basis of molecular biology and genetics. Moving forward, DNA replication. It is basically the process of copying genetic material before cell division. It ensures identical DNA is passed to daughter cells. It occurs during the S phase of cell cycle, which follows the semi-conservative model of DNA replication. Replication is bidirectional and it occurs in 5' to 3' direction. It begins at a specific site called as origin of replication. It is a reaction in which daughter DNAs are synthesized using parental DNAs as the template, transferring the genetic information to descendant generation with a high fidelity. What are the characteristics of replication? It is semi-conservative replication, bidirectional replication, semi-continuous replication, and it has high fidelity. Now we are going to see the enzymes and proteins which are involved in replication. DNA polymerase. It matches the correct nucleotides that joins polymerizes adjacent nucleotides to each other. Helicase. Helicase basically unwinds the DNA and it melts it. Next one is primase. It provides an RNA primer to start polymerization. Now what is SSB proteins? It keeps the single strand of DNA after it has been melted by helicase. What is gyrase? Gyrase is a topoisomerase that relieves the torsional strain in DNA molecule. Then we have ligase, which joins adjacent DNA strands together. And the last one, telomerase. It finishes off the DNA strands in eukaryotes. Now we'll see DNA polymerase 1. The first DNA-dependent DNA polymerase, which is DNA polymerase 1, was discovered in the year 1958 by Arthur Kornberg, who received Nobel Prize in Physiology and Medicine in the year 1959. DNA polymerase is also considered as Kornberg enzyme. Later, DNA polymerase 2 and DNA polymerase 3 were identified. All of them possesses the following biological activity. It has 5' to 3' polymerase activity and it also has exonuclease activity. DNA polymerase 1 is mainly responsible for proofreading and filling the gaps, repairing the DNA damages. DNA polymerase 2, it is a temporal functional when DNA polymerase 1 and DNA polymerase 3 are not functional. But it is still capable of doing synthesis on the damaged template. It participates in DNA repair process. Next one is DNA polymerase 3. It is a heterodimer enzyme composed of 10 different subunits, having the highest polymerization activity. The true enzyme responsible for the elongation process is DNA polymerase 3. Then we have primase. Now, RNA primase is able to synthesize primers using free NTPs as substrate. Primers are short RNA fragments of several nucleotides long. Next one is helicase. also referred as DNA B. It opens the double-stranded DNA with consuming ATP. The opening process with the assistance of DNA A and DNA C. Next one is SSB protein or single-stranded DNA binding protein. Now SSB protein in the single-stranded form of order, it maintains the DNA template. It prevents the double-stranded DNA formation. It protects the vulnerable single-stranded DNA from nucleases. Next one is DNA gyrase. It cuts the phosphodiester bonds on both strands of the DNA. It releases the supercoil constraint and reforms the phosphodiester bond. It can change the DNA into negative supercoil state with the consumption of ATP. Next one is DNA ligase. Ligase connect two adjacent single-strand DNA strands by joining the 3'OH group of one DNA strand to the 5' group of the another DNA strand. Sealing the neck in the process of replication, repairing and recombination and even splicing is where DNA ligase performs its function. Now, in the summary, we can see the different enzymes involved in DNA replication. First one is DNA helicase, which unwinds the double-stranded DNA by breaking hydrogen bonds between complementary base pairs. It creates a replication fork. Then we have topoisomerase, which are DNA gyrase in prokaryotes. Now, this enzyme relieves torsional stress in supercoiling that develops ahead of the replication fork by cutting and rejoining DNA strands. Then we have single-strand binding protein. SSB proteins binds to single-stranded DNA, preventing re-annealing and protecting the strands from degradation. Next one is primase. Primase synthesizes short RNA primers that provide a free 3-OH group necessary for DNA polymerase to begin DNA synthesis. DNA polymerases are the key enzymes responsible for synthesizing new DNA strands. In prokaryotes, DNA polymerase III performs most of the DNA synthesis, while DNA polymerase I removes RNA primers and it fills the gap. While in eukaryotes, DNA polymerase alpha initiates the synthesis. Then we have RNase H. RNase H removes RNA primers from RNA DNA hybrids during lagging strand synthesis. DNA ligase, as we have seen, it joins the Okazaki fragments by forming phosphodiester bonds. It ensures the continuity of the DNA strand. Then the last one is telomerase in eukaryotes. It extends the telomeric ends of chromosomes, preventing loss of genetic material during repeated rounds of replication. So this is it for the video. We'll meet in the next video. Thank you.